Hi everyone, this is from Vivid Botany. We are looking the NEET 2021 paper. Here we have again mass of the following question. That list 1, list 2, protoplast fusion. Protoplast means the cell without cell wall. Protoplast is cell without cell wall is called protoplast. So here in this process, two different cells of two different plants are taken together in a container. For example, we have one example in textbook about this is that one cell that we take from potato, let us take this cell, we take it from potato. Another cell that we take it from tomato. We know that potato is Solanum tuberosum. Solanum is genus name. Tomato is the Lycopersicon. So since the two cells are plant cells, they have cell wall. So to remove the cell wall, they make use of PEG. That is, first the cell walls are removed by cellulase, hemicellulase, pectinase, like that. So by cellulase, hemicellulase, the plant cell walls are digested. After removing the cell wall, the protoplasts are found and two protoplasts are joined together with the help of PEG, that is polyethylene glycol. So when two cells are joined together, it will form a new cell that is having the chromosomes of both the cells. So this is a hybrid cell. We call it as somatic hybrid. And this is botanically called as Pomato. It's a combination of both the words potato and tomato, and people also call it as Solanopersicon. So Solanopersicon, scientifically a big success in the history of biotechnology, but it failed to attract the taste of common public. Public could not like this product for a long time. It is a failure. So for protoplast fusion, here we can give that example as Pomato, that is potato plus tomato. One more thing is that since potato, tomato both are from different genera, because potato is Solanum tuberosum, tomato is Lycopersicon esculentum. So both are since from two different genera of same family, it is called as intergeneric hybrid. It is called intergeneric hybrid. That is the hybrid obtained from two different genera of same family. So it's called intergeneric hybrid. It is also called as parasexual hybrid because it is obtained from without sexual reproduction. So we also call it as parasexual hybrid. We call it as intergeneric hybrid. Intergeneric hybrid means two different genera are involved in making out of the pomato. Then another is plant tissue culture. We know that in plant tissue culture, we take what it is, an explant. An explant is part of the plant taken from tissue culture. It is cleaned by detergents and then the distilled water is taken. It is made into the nutrient medium. This nutrient medium, the explant is added with inoculation, that is completely in aseptic conditions like in laminar airflow chamber, and sterilization of nutrient media is also done in autoclave manner. And plant tissue culture, so many scientists are involved here, especially from India. We have that Maheshwari, Panjan and Maheshwari was very much involved in the plant tissue culture. And we have here in plant tissue culture totipotency, a word is there. Totipotency means it's called cellular totipotency. That is, every part of the plant is able to generate to give rise to a new plant with provided suitable conditions is called as totipotency. This was discovered for the first time by G. Haberland. G. Haberland, and he was the person who for the first time coined the word also. He coined the word. The word was coined by the same person, that is G. Haberland. And next, that is Meristem culture. Meristem culture, by which we can obtain the virus-free plants. How? Here, where meristem is present generally in a stem from axillary bud and apical bud. The bud contains the meristem. We know that bud is condensed part of the stem that is overlapped by young leaves. Inside the bud we find the meristem. The meristem, the cells which are actively dividing throughout their life, they are rich with energy, rich with mitochondria, rich with ATP. So they do not allow any inoculation or incubation of the virus inside. So they are non-vasculatured, they don't have the xylem and phloem vasculature, and they are free from viruses. So when you take explant from this meristem, you can obtain virus-free plants. It was for the first time done by, that is virus-free plants. Virus-free plants from Meristem culture. Meristem culture includes here axillary bud and apical bud. We know that axillary bud is also called as lateral bud, and apical bud is also called as terminal bud. Apical bud is helpful in vertical growth of the plant. Axillary bud is helpful in lateral growth of the plant, that is horizontal growth of the plant. So this virus-free plants from meristem culture is done by the first time by two scientists, then is Morrell and another one is Martin, Martin, in 1952. And apart from that, tissue culture was involved with so many scientists like Murashik, Skoog, Miller, White, Nietzsche, Johari, Maheshwari, Sharp. All these are involved in the tissue culture. So we can write here that meristem culture is virus-free plants, that is here option 4. So definitely this is second option. Let us see here A2, that is protoplast fusion to Pomato. Already I got it here. Then B1, that is plant tissue culture, is based on totipotency. Totipotency means what? The ability of plant to give rise to a new plant under suitable conditions is called as totipotency. We don't find totipotency in animals. It is exclusive character of plants. It is defined as ability of any part of the plant, to give rise to a new plant is called as totipotency. So the wholesome tissue culture process is based on the totipotency principle. It is called as cellular totipotency nowadays. So every cell, if you take the microspore, that is pollen grain, you can obtain here that plants that are haploid plants can be obtained, that is called as androgenesis. You can obtain plants from ovule like pollen grain, seed, root tip, shoot tip, any part of the plant can be obtained. And meristem culture, we can obtain here virus-free plants. Remember, not only virus-free plants, but also you can obtain here free from mycoplasma, fungi, bacteria also can be obtained by the meristem culture. For example, a plant is so beautiful having desired characters but that is having infected by different kinds of pathogens. Now, from that infected plant, can we develop a pathogen free plant from that? Yes, it is possible. If you take the meristem of the plant as the explant, you can develop virus free plants, pathogen free plants from that infected plants. Remember one thing, that the meristem is not of the root but of the shoot. Only the shoot apical meristem, that is the axillary bud, and the terminal bud, can be taken as explant from which we can obtain the virus free plant. So this is a novel technique very much exploited in the tissue culture process. Then micropropagation. Micropropagation is something that that is tissue culture process. Here is that in this process, when you take explant of same plant in different test tubes, every explant will grow into an individual plant. All these plants, since are taken from same mother plant, they have same phenotype and same genotype. So formation of group of plants which are phenotypically, genotypically same to that of their mother plant is called as somaclone. Soma means vegetative, clone means the similar group of plants. So somaclones are obtained from micropropagations. If any microscopic variations are there in somaclones, such are called as somaclonal variations. Sometimes somaclonal variations also become the basis for selection, that is called clonal selection. So here we see that protoplast fusion, Pomato is an example, Lycopersicon, it's called Solano persicon. I wrote it somewhere, this is called Solano persicon. So Meristem culture, that is virus free plants, and micropropagation, that is somaclones, and plant tissue culture, that is totipondency. So here this question is entirely built from only subtopic, that is tissue culture, of one chapter that we have in +2. So we should have here, we should know the Pomato, Solano persicon, totipotency, somaclones, somaclonal variations, virus free plants. Then only you can keep it the right one. Hope my video, my explanation is very much useful to you. To subscribe my channel, we are talking from Vivid Botany. I will be giving you thorough guidelines as far as botany is concerned for NEET. I will be giving you excellent guidelines for NEET 2022. So subscribe my channel and let us crack the NEET 2022. Thank you.